Unit 4, Lesson 2, The Chromosomal Basis of Inheritance. BioExpert video lecture. This video lecture deals with 4 learning targets. How to analyze data sets mathematically to determine Mendelian patterns of inheritance. Explaining how the chromosome theory of inheritance connects the physical movement of chromosomes to Mendel's laws of inheritance. Relating the alteration of chromosome number to genetic disorders. And analyzing sex-linked patterns of inheritance. Mendel's laws of segregation and independent assortment reflect the rules of probability. When tossing a coin, for example, the outcome of one toss has no impact on the outcome of the next toss. In the same way, the alleles of one gene segregate into gametes independently of another gene's alleles. The multiplication and addition rules of probability can be applied to monohybrid crosses. The multiplication rule states that the probability that 2 or more independent events will occur together is the product of their individual probabilities. Probability in an F1 monohybrid cross can be determined using the multiplication rule. Segregation in a heterozygous plant is like flipping a coin. Each gamete has a half chance of carrying the dominant allele and a half chance of carrying the recessive allele. The addition rule states that the probability that any one of 2 or more exclusive events will occur is calculated by adding together their individual probabilities. The rule of addition can be used to figure out the probability that an F2 plant from a monohybrid cross will be heterozygous or homozygous. We can apply the multiplication and addition rules to predict the outcomes of crosses involving multiple characters. A dihybrid or other multi-character cross is equivalent to 2 or more independent monohybrid crosses occurring simultaneously. In calculating the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied. We now know today that Mendel's hereditary factors were actually genes. We can show that genes are located on chromosomes. The location of a particular gene can be seen by tagging isolated chromosomes with a fluorescent dye that highlights the gene. Mitosis and meiosis were first described in the late 1800s. The chromosome theory of inheritance states Mendelian genes have specific locations or positions on chromosomes, and chromosomes undergo segregation and independent assortment. The behavior of chromosomes during meiosis can account for Mendel's law of segregation and independent assortment. Here we see a cross between 2 purebred plants for the color and texture of seeds. The plant on the left has the dominant traits, or the dominant alleles for both genes, and the plant on the right has the recessive allele for both genes. And notice there are 2 possible gametes that can be created, one from each parent. Parent on the right can produce gametes that have yellow and round alleles, and the gamete on the left has gametes that produce green and wrinkled alleles. If we take 2 of the round and yellow seeds produced by the F1 generation from these parents, the F1 generation are all hybrids or heterozygous. Once we introduce heterozygous genotypes, the number of possible gametes increases by a certain factor. And that results in a rather unique ratio that can illustrate the law of segregation and law of independence. We know that in humans and some other animals there is a chromosomal basis of sex determination. In humans and other mammals there are 2 varieties of sex chromosomes, a larger X chromosome and a smaller Y chromosome. On the ends of the Y chromosome, there are regions that are homologous with corresponding regions on the X chromosome. For example, the SRY gene on the Y chromosome codes for a protein that directs the development of male anatomical features. Females are XX and males are XY. Each ovum contains an X chromosome, while a sperm may contain either an X or a Y. Other animals, as shown here, have different methods of sex determination. A gene that is located on either sex chromosome is called a sex-linked gene. Genes on the Y chromosome are called Y-linked genes, and there are very few of these. Most genes that are sex-linked are on the X chromosome and are called X-linked or sex-linked genes. X chromosome has genes for many characters unrelated to sex, whereas the Y chromosome mainly encodes genes related to sex determination. X-linked genes follow specific patterns of inheritance. For a recessive X-linked trait to be expressed, a female needs 2 copies of the recessive allele. She needs to be homozygous recessive. This is because she has 2 X chromosomes. A male, on the other hand, needs only 1 copy of the allele. He need only be hemizygous for the recessive trait. This is because male mammals have only 1 X chromosome. X-linked recessive disorders, for this reason, are much more common in males than they are in females. Some disorders caused by recessive alleles on the X chromosome in humans include color blindness, Duchenne muscular dystrophy, and hemophilia. Other genetic disorders in humans result from alterations of chromosome number or structure. Large-scale chromosomal alterations in humans and other mammals also often lead to spontaneous abortions or miscarriages, or they may cause a variety of developmental disorders. Plants are able to tolerate such genetic changes better than animals do. Abnormal chromosome number can result from non-disjunction. In non-disjunction, pairs of homologous chromosomes do not separate normally during meiosis. As a result, one gamete receives 2 of the chromosome, and another gamete receives no copy. Aneuploidy results from the fertilization of gametes in which non-disjunction occurred. Offspring with this condition have an abnormal number of a particular chromosome. A monosomic zygote has only 1 copy of a particular chromosome. A trisomic zygote has 3 copies of a particular chromosome. Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. Triploidy, 3N, is 3 sets of chromosomes. Tetraploidy, 4N, is 4 sets of chromosomes. Polyploidy is common in plants, but not in animals. Polyploids are more normal in appearance than aneuploids. There are other possible alterations of chromosome structure. Breakage of a chromosome can lead to 4 types of changes in chromosome structure. Deletion removes a chromosomal segment, which actually involves removing a gene or set of genes. Duplication repeats a segment, which involves repeating a gene or set of genes. Inversion reverses the orientation of a segment within a chromosome, so genes are displaced in their location along the same chromosome. And translocation moves a segment from one chromosome to another. So one set of genes on one chromosome is swapped for a set of genes on a different chromosome. Human disorders due to chromosomal alterations often result. Alterations of chromosome number and structure are associated with some serious disorders. Some types of aneuploidy appear to upset the genetic balance less than others, resulting in individuals surviving to birth and beyond. These surviving individuals have a set of symptoms or syndrome characteristic of the type of aneuploidy. Down syndrome is an aneuploid condition that results from 3 copies of chromosome 21. It affects about 1 out of every 700 children born in the United States. The frequency of Down syndrome increases with the age of the mother, a correlation that has not been explained. Non-disjunction of sex chromosomes produces a variety of aneuploid conditions. Klinefelter syndrome is a result of an extra chromosome in a male producing XXY individuals. Monosomy X, called Turner syndrome, produces XO females who are sterile. It is the only known viable monosomy in humans. Finally, disorders can be caused by structurally altered chromosomes. The syndrome, Cri-du-Chat, cry of the cat, results from a specific deletion in chromosome 5. A child born with this syndrome is mentally retarded and has a cat-like cry. Individuals usually die in infancy or early childhood. Certain cancers, including chronic myelogenous leukemia, or CML, are caused by translocations of chromosomes.